Well, thank you all for being here, and thanks to the organizers for inviting me. I think it's a great idea to try to get cross-fertilization and discussion through these fields that I've definitely thought a lot about myself, but not the way many of you do. Sorry for the cut-off slides. We're going to have to live with it, I guess. So I'm going to talk to you about decision-making and give you a little bit, since it's a very diverse audience, give you some background on choice reaction time and a little bit of confidence. I'm going to then talk about alternatives to evidence accumulation models and try to provoke a little discussion about a very different way of thinking about information processing than most of you from the machine learning and neural network architecture think about. Okay, so a decision is a commitment to a proposition among alternatives that arises through deliberation. The neural mechanisms, I think, give us a window on cognition, and I use this word cognitive to imply operations that are more elaborate than simple sensory motor programs and that unfold on a flexible time frame. And an example might be trying to decide to take the left or the right path here, that because that invites evaluation, what the psychologists call prospection, that is using your knowledge based from memory stores about what might be at the end of these paths, strategizing about whether or not how far to go perhaps before you could turn back and change your mind, and planning. And it has an origin, I think, in animals, and therefore is amenable to neurobiological study in foraging and exploration. And I think that's the only natural aspect of the tasks that we study that relates to natural behavior. Now, I'm interested in this problem not because of interested in decision-making per se. I mean, my goal is not, say, to make traders, criminals, better at what they do, but rather to understand how the human brain works and how it establishes cognition. And that's why this particular, which is a bit parochial definition of decision-making, interests me. It's not one that might apply to a fly or a roundworm or something like that also make decisions, but that don't operate on flexible time frames and so forth. So we use the monkey model to study very complicated decisions in an animal. Ultimately, I'd like to unravel some of those mechanisms in the currently genetically tractable model system, the mouse, and then, of course, it bears on the how our human brain works. I think that matters a great deal if you come to biology from the perspective of medicine, as I do. Okay, so the task that we've used in the lab for many years, and I apologize to those that I'm boring with this, is a random dot kinematogram, a random dot motion task in which an animal must decide the net direction of these randomly flitting about spots, these dots. And we control the difficulty of whether of this decision, right or left, by controlling the probability that a dot plotted at one moment in time will be displaced at another very soon moment in time. And that probability we call the motion strength or the motion coherence. We also control difficulty in some experiments by controlling the viewing duration. But in the experiments I'm going to tell you about right now, the monkey controls the viewing duration by telling us his answer with a fast eye movement. We call that a saccade, as soon as he's ready to make an answer. And over about 15 years, we think we understand how this process works in the brain. And the idea is that evidence, noisy evidence from the visual stimulus in this case, transduced by neurons in the visual cortex, which are also noisy, give rise to what I'll call the momentary evidence, the things that are the bits of evidence that come from the world and are changing moment by moment in this dynamic display. We know that that transduction takes place in direction-selective neurons in the visual cortex, this area known as V5 or MT, where many neurons, really 90-something% of the neurons, are direction-selective and organized in this sort of columnar fashion, so that the momentary evidence is the difference between a noisy rightward and a noisy leftward signal. And that difference is represented in this Gaussian on its side here, since this motion is to the right. Hopefully you can see that motion is to the right. If you can't, talk to me later. I'm a neurologist. This motion is being transduced by rightward and leftward preferring neurons. The subtraction is the momentary evidence. And the idea is that neurons with longer time constants in the brain, they represent the accumulation of that momentary evidence. Okay, so this represents a random walk or a path of diffusion, and when that accumulation reaches a critical level, say this gray up here, this upper bound, the animal would choose right. If it reached this, the animal would choose left. Okay, and the idea about these bounds is that, or actually I should say the beauty of this simple idea is that it unites in a common framework the accuracy of a choice and how long it takes to make that choice. Okay, and the intuition there is that if the bounds were closer to the origin, then this trial would have ended in a leftward choice, which would have been an error, but at a savings of time. Now, in our hands, if you ask monkeys to do this task, and you plot their reaction time as a function of motion strength, your reaction time is the time that elapses from onset of the random dot motion to the beginning of the monkey's eye movement response. And this motion strength axis is signed so that positive numbers, like here, mean strong rightward motion. This positive big negative number means strong left motion. So the most difficult cases are in the middle of the graph, and you can see the reaction times are longest there. And we can explain this pattern of mean reaction time as a function of motion strength with this very simple equation. It involves these terms b for where the bounds are, as I just told you. It involves one other coefficient that multiplies motion strength, shown here by uppercase C, and this constant just multiplies that into units of signal to noise. And it has this other term, which is we call the non-decision time. We measure reaction time, but this mechanism only accounts for the actual time of accumulating the information. Okay, so the sum of that and non-decision time gives you your reaction time. Now, the nice thing about this, as I said, is that this mechanism should explain in one pass the simultaneous, I meant to say, it should explain both the choice and reaction time. So this fit predicts what the choice function should be, the proportion of rightward choices as a function of this same motion strength axis. And the idea is because this function is a logistic function of this term, which has the coherence, the motion strength in it again, but also this term K and B, the bound, and this term that converts motion strength into signal to noise. And having established what those terms are, by fitting the reaction times, we're in a position to predict what the choices should be. So those are the predictions, and these are the data, and they look awfully much like fit. And I would have said until meeting Gabe Stein, who's a new student in the lab, that this was like sealed the deal for me, that there's really no alternative to bounded evidence accumulation as a model, but I'm going to have to do a little bit of a mea culpa in a few minutes. And so keep me honest on that, because there's going to be a lot of seemingly aggressive statements as well. But most of that aggression in this case is going to be at myself. So I should say we didn't invent this model. It was introduced during World War II by Abraham Wald, who actually did the mathematical work here at Columbia. He was a refugee from Austria. Alan Turing used this kind of a bounded evidence accumulation as part of the code-breaking work in World War II. And these are sort of the main pioneers and current champions of this kind of a model. Okay, and this is not an exhaustive list. It's become a very popular model, but I did not invent it. I don't want you to think I did. Okay, so now in the brain, it has a beautiful neural correlate, and actually in many brain areas, and I'm going to just introduce one of them, which is the lateral intraparietal area. This is the monkey brain, and this MRI is taken at this slice here through the posterior parietal cortex. And this area, LIP, is part of what we call Brodmann's Area 7, the posterior parietal cortex. It's the part identified by Richard Anderson that projects to the frontal eye field and the superior colliculus. So its output is influencing structures in the brain that control eye movements. And for that reason, many years ago, Bill Newsom and I thought we could study these neurons to understand decision making. And so I'm going to show you the main property these neurons have that make us think that they are the neural basis of what might be called, I'm going to try to convince you at the end of this, epistemic state, a sense of knowledge, a sense of thinking, of committing to a proposition. Okay, Nancy Canwisher said, I don't know what a thought is. I'm going to explain what a thought is. Okay, so, all right, so This is the simplest experiment in the world. This is our screening experiment. It's called working memory task or oculomotor delayed response task. Monkey stares at a fixation point, sees a briefly flashed spot of light. That's where it was. I'll call that region of space the response field of the neuron in a second. And we're going to listen to this neuron. I hope you can hear this. As the monkey is sitting there waiting for the go signal to make an eye movement to the remembered location of that briefly flashed spot. All right, great. Nothing like the sound of a neuron on a Wednesday afternoon. Okay, so this is what I meant by persistent activity. These tick marks are the times of the spikes, the action potentials from the neuron, played over a loudspeaker. This represents the onset and offset of the red spot. This is the cue for the monkey to make his eye movement. This one's when he actually made it, okay? That's persistent activity. I want to point out something semi-philosophical, but I think really important, is that this is the essence of what allows us to have cognition, is persistent activity. It is a ubiquitous feature of neurons throughout the association cortex. It confers what I call freedom from immediacy. Because if you think about it, any interesting mental act relies on the ability to compute and represent information in a time frame that is free from the immediate changes in the world and the need to control body musculature in real time, okay? And that I think in evolution, it was this emergence of persistent activity that made us sort of non-reflexive and give us contingency in the context of decision making that is the key to understanding the emergence of cognition. Now, in LIP, because it projects to an area of the brain that cares about space, if that spot is flashed to the left, as it was in this example, instead of seeing an elevation of activity, there's a depression of activity, or at least no change. And that's the modifier here, that in this case, the persistent activity is spatially selective. So what we do is, in the lab, is we contrive tasks that take advantage of these neurons. We ask the monkey to make a decision about a target that's in the response field of the neuron and another target that's outside it. And in this case, he's deciding about whether motion is to the right or to the left, okay? Again, nothing natural about this at all. It's exploiting a feature of the neuron in order to study something interesting, which is how is it that the brain assembles the evidence to reach a decision. Now, there's nothing interesting also about the fact that this neuron is capable of jumping to a rate that says I'm going to make this eye movement, or not jumping, maybe jumping down or something, to say to indicate the monkey's going to make that eye movement. But in fact, that's not what the neurons do. And so here is, because when you study neurons in the setting of this reaction time experiment, we can focus in on the period of time that the monkey is forming his decision, but hasn't yet committed to a choice. And that's what the thesis work of Jamie Reutman was many years ago. You can't see the papers, unfortunately, down here. If we look at the average firing rate plotted aligned to motion onset, and we look at the trials that are going to end in a rightward eye movement, an eye movement to the target in the response field, not surprisingly, the firing rate increases. If he makes the opposite eye movement, the firing rate decreases, okay? So that's obvious in a sense because we selected the neurons on the basis of that property. But you see that this rise and fall is kind of gradual, and it's dependent on the motion strength. So these three curves are the strongest, gold, the weakest, the zero, this blue, the monkey might as well be guessing, but he doesn't know that because there's a lot of difficult cases. And then here's in red one of the intermediate motion strengths. So you can see that this neural response is indicative not just about what eye movement is going to be made, but something about the quality of the evidence leading to that choice. This is a reaction time experiment, and these trials are stopped way before the eye movement. I'm stopping them in the plotting. That they're way before the monkey actually makes his eye movement, okay? So, of course, you can look at the responses backwards in time from the fast eye movement, the saccade. So here's zero there. And now you can see that these responses, even though they began growing or meandering at different rates, they reach a common level up here. Not so true down here. Okay, so, and if we look closer at this, we get a better clue by looking at all the trials, all the motion strengths, and just group them by reaction time. So So these are those same trials, but with all the motion strength included, now grouped by quantiles of reaction time. So you see very fast reaction times, there's a rapid rise. Very slow reaction times, there's a very much slower rise. You see this little dip and rise in all of these traces. But the feature I want to draw your attention to is that the responses kind of reach a common level, something that kind of looks like a threshold, about 100 milliseconds before a monkey moves his eyes. And that, we think, is a signature of that threshold or bound on the decision, okay? And so we think that threshold is detected by neurons downstream of LIP and all of the other areas that look just like LIP. And now I would also say that if we take that 100 milliseconds and pay attention to when the information from the world, which is at time 0, began to affect the LIP responses, that's another 200 milliseconds roughly. It's 190 or so. And that happens to correspond to within maybe 10 or 20 milliseconds to that non-decision time term when we fit the reaction times. So that's kind of a nice neural correlate of those very, in some ways, mysterious delays, especially this one. Now, I want to make a point, and it's a bit of a critical point, but it's extremely important, I think, to many of you. It's about decoding. If you look at the neural response, say just when the responses are beginning to exhibit in the effects of stimulus information at time 0. That's out here. So just after the responses begin to diverge by choice and motion strength, and you were to say apply a GLM to it or a machine learning, some decoder of some kind, you might write a paper that says that actually the decision, there's enough information here to decode what the monkey's going to do at that point in time. And there are many papers that are written like that, okay? And, but the fact of the matter is, the nice thing about reaction time experiment is that we know that the monkey didn't decide that. All right? So there's a lot of things, I'll just warn you, that there's a whole literature about rapid detelling of faces and animals from other things in the world and so forth. It's all based on that basic error, and we all make it, I'm guilty of it too, of assuming that our decoding is what the brain is doing. So this is a bit preachy, and I apologize for that. But we shouldn't confound the experimenter's decoder or inference with that of the brain. We should test those things. Now, there's another trick that we use in our science, and that is to, if without looking at the neurophysiology, is to look at what we call psychophysical reverse correlation, or well, I'll abbreviate that as psychophysical kernel. And that is to take the information from the movie, transform it by the process of extracting motion information for right and left, okay, which I'll call motion energy extraction, and then average that in accordance with the choice. So we get what, in arbitrary units, support for the choice that the animal is going to make when he's basically guessing, when he's looking at a pure noise stimulus. In fact, it's information in the stimulus that at least is driving some fraction of his binomial variance. And you can see that, and that's what's shown in green here, okay? You might be tempted to think that there's a delay here, but there really isn't, because the extraction of the motion information is itself this is its impulse response, has a delay in it. And so we conclude that basically this is from human data, it so happens, that the human was using all of the motion information from right at the beginning of the trial, okay? Now, the nice thing about this is that it gives us another handle on this non-decision time. Because if we ask what does the human use the information at the end of the trial, this is now aligned to the subject's choice. This was work in collaboration with Daniel Walpert where subjects used their hands. So their non-decision time was about 450 milliseconds, depending on which subject it was. And so we can use this to recover the non-decision time. So if we make a line here, this was the mean for this 430 milliseconds. We make a line, we say effectively, at least on average, that the subject could have stopped accumulating information at using information from this point forward. And of course, we also estimate its standard deviation in order to explain, say, distributions of reaction times. So we can blur that out a bit by that standard deviation, and then deconvolve with this kernel, and lo and behold, what we end up with is a nice confirmation or corroboration of the non-decision time in these experiments inferred. And of course, this experiment was all about why we use this extra the information in the world to change our minds. And so that's a lot of time, actually, that a stimulus is shown where there was extra information after the choice was made. I want to say one more thing in this sort of introductory section, and that is that the same basic framework can explain the confidence that we have in a decision. So confidence, you can think of it like a belief. It's a good operational definition of confidence is the probability that my decision I'm about to render before the world gives me a real answer is correct or incorrect. And so what is confidence in this framework? Well, basically, it's a mapping between the state of accumulated evidence and the probability that a decision rendered on that evidence will be correct. And if you do the math on that, it has a very interesting structure. So this is state of evidence on the vertical axis, and this would be time taken during the decision, and or maybe the time supplied by the experimenter if the experimenter controls the viewing duration. And you notice that the lines of the ISO probability of being correct lines, okay, we'll call them ISO confidence lines, are curved. They're time dependent. In other words, to be concrete about it, if you accumulated this much evidence in this much time, you would know that on average you should be correct about 75% of the time. But if you accumulate the exact same amount of evidence and it took you this long to get it, then you're only going to be correct about 65% of the time. Okay. Now that's the reason I want to share that with you, is that many of us in this room probably apply normative theory and Bayesian rules to update information towards beliefs. But this implies that when the brain does that, it ought to at least do that dynamically. That is a fixed prior, a fixed asymmetric prior. Say motion is more likely to be to the right 75% of the time or something. You know, that should affect decisions dynamically as a function of time, okay? But most of us don't think about that because the prior itself is fixed. It might be the same prior for thousands of trials in the case of a monkey, and yet should affect information gathering or accumulation of information dynamically. Okay. Now, I'm going to turn to the next part, and this I'll apologize in advance for being a little bit preachy, but remember, part of this is directed at myself. So, and that's not the lovey-dovey part of the thing. That's more the warrior part. With you guys, it'll be lovey-dovey. Okay. So here's a rendering again of integration or accumulation in general. The name of the models doesn't matter. It's sequential sampling with optional stopping, and it's in a very simple framework. Now, why should we care about this? The neurobiology only makes sense. This is going to be a problem for me if these slides go down like that. This neurobiology only makes sense if we think the animal is integrating. We are interested in decision-making as a window on cognition. Cognition meaning that there must be several samples. Decision-making implies the contingency already. So many samples, it's something evolving in time to reach a decision. So what are the alternatives and how can we tell? Well, the simplest alternative, which I'll just do very quickly, is that the integration itself is leaky. And that's not I don't think that's that important. It happens not to be true in this case. And how do we know that? Well, the one way to test it is to vary the stimulus duration. In other words, don't do reaction time. Let the experimenter control the viewing duration. And if you do that, signal to noise should improve by the square root of time if integration is not leaky, if integration is perfect. And so here's an example of that this is work from Rusbek-Kiani. So here's plotting the discrimination threshold, that's inverse sensitivity, as a function of the the experimenter-controlled duration. These are both on log axes, if you look closely. And this line is the square root line. It's the power minus 1/2 line. And so, at least for up to about 500 milliseconds or so, the signal to noise is improving by square root time. And we think that this performance is limited by a bound and not by a leak. Why? Because we can apply an impulse. We can look at long duration trials and apply a subtle perturbation to the stimulus. And what we find is that if you apply the subtle perturbation to the stimulus late in time, it doesn't affect the choice. But if you apply it early in time, it does. Now, if it was a leak, if there was a leak to the integration, then it's the early information that leaks away, and the late information should have affected the decision. And so, but the idea is that when we watch something, when we make a decision, and we even though we have more information in the world that we could use and perform better, that the brain times out. It basically is performing reaction time, even though there's no overt manifestation of a reaction time. And that's really important for a variety of reasons. The one I want to emphasize with you is that it is not optimal, and it makes you wonder about being too invested in normative solutions. So to this I say normative schmormative. So in other words, you wouldn't know this unless you measured it, but there's no rationale whatsoever for stopping the accumulation early if one's goal is to achieve reward, and that is indeed the monkey's goal. Now, one could argue, I guess, in a circular way, and I agree with this argument, that there's a way to sort of justify this, which is that maybe there's sort of an energy cost to making the decision at all. But it's a very subversive idea that the brain is essentially using or applying a speed accuracy trade-off when there really is no speed measurement. And I would submit to you that most of perceptual learning is not so much accomplished by changes in synaptic weights, but accomplished by changes in strategy of things like speed accuracy trade-off under the hood, not manifested in any obvious way other than performance improving. Okay, now here's the other alternative that I've been concerned about lately, which is that what if there's no integration at all? That would really be a bummer for us. So this model says, here comes a sample, compare it to a threshold, which I'll use the same B for bound, but just a threshold now. If you get there, stop and decide right or left. But if you don't, then don't integrate anything, just get another sample, okay, until you do, okay? In signal detection theory, this used to be called something like probability summation, but the idea is that there's no memory, there's no integration going on. It's sort of to capture that class of models, okay? It's sequential sampling with optional stopping, but no integration. All right, so now that model predicts a few things that are interesting to look at. One is that the psychophysical kernel, that reverse correlation, that shows us the information, the stimulus that affects the choice, it should be aligned to the response, okay? So in other words, if this is when the animal or the human makes the choice, there should be a bump in the leverage of information at some fixed duration, spread out, by things like non-decision time and so forth. But it should be at this point in time, and going back in time, it should vanish. It should not affect the choices. The information in the stimulus should not affect the choices if that information was presented and the choice wasn't made. Okay, and just to compare this, I showed you this kind of a function in green before, but in a bounded integration type model, there's leverage of the stimulus information on the choice way back from the beginning of integration, of course. What else can we do? Well, we can also ask whether, sort of in the same vein, we predict that early perturbations on the stimulus should not affect later choices. And when Alex Huck was a postdoc in my lab, he performed an experiment where he put textures, little pulsy textures in the background of random dot motion, and he subtly affected the monkey's choices. And this shows that a little pulse of motion presented behind random dots at between 0 and 100 milliseconds has a lasting impact on the animal's both choice and reaction time right on out to almost a second. Okay, so that's incompatible with this extrema model and is support for integration. Now, here comes the mea culpa. It turns out that this model, given the degrees of freedom it has, which are very few, can also explain the choice and reaction time. Now, I didn't believe this. I thought it was impossible for an extrema model to explain choice and reaction time, but the fact is it can. So there's a red and a blue curve here. One is a fit of the bounded integration model, one's a fit of extrema, and you really can't tell them apart. So that's the mea culpa. On the other hand, if you have an animal or a human and you measure them in both a reaction time experiment and you measure them in an experiment where you control the viewing duration and get that square root improvement up to some point of a bound. I've now reversed the plot and plotted this as sensitivity instead of threshold. These are the data you actually saw before, and these are data from a monkey performing the reaction time experiment, the same monkey. And you can see that you can predict that in red, we're able to predict the monkey's improvement as a function of experimental controlled duration. We're able to predict his performance from the data that we gather in a reaction time experiment. And we can't do that with when we try to fit the reaction time with the extrema and predict the same animal's improvement as a function of time. That's a complete miss. Okay, it's rare that one would have that kind of data from an animal, but of course you can gather that kind of data from a human. Now, the third alternative model that I want, and last, that I want to talk to you about is what I call random snapshot. And it's not necessarily random. The idea here is, this is not a sequential sampling model. The idea here is that the animal, either because of some arbitrary time during the stimulus presentation, or basically maybe some event in the world, maybe some cluster of clicks or a bright flash or something, causes the animal to just take a sample and then compare this to a threshold. Positive, choose right; negative threshold, choose left. Or there's going to be no more samples here, guess. So that mechanism, I think, is worth considering, and it would be, again, a bummer if it was to hold. Obviously, one way to exclude this is to perform a reaction time experiment. But there are many papers in the literature that assume integration but never measure it with reaction time. So, and this is important to me because there are, if one is to for example, make conclusions about activity in LIP or MIP or frontal eye fields or many of the areas that show this kind of activity, it's worth knowing whether or not one actually is looking at integration. So I should say that the nice thing about this kind of a mechanism is it can explain any psychophysical kernel, because we could just, we could match any kind of weighting of early and late information to saying that, well, that's when the animal happened to look at the stimulus. Okay, so I'm going to just give you an example from the literature that caught my attention. So this is from a paper by Yates et al. That came out this year. They call this motion task a pulse experiment. There are 7 pulses, what are these little hexagonal circles are. This is a movie of 7 pulses, each lasting 150 milliseconds, and each pulse consisting of 19 elements, Gabor elements, little tiny patches of grating. And the idea is that in any one pulse, some fraction of that 19 elements is moving, and those that are moving are all moving in the same direction. But it's not the same number in every one of these 7 pulses, and in fact, in the difficult cases, maybe this pulse would have had some leftward motion in it. Okay, but all the elements within a pulse are moving, and they're moving in a way that if you were looking at them, you'd never mistake their direction, which is what made me start thinking about this kind of an alternative model. So here's what the data look like. This is a really good cautionary tale, I think, for us, is that this is the proportion of right choices as a function of motion strength. But motion strength itself is a description that assumes integration. So what they do is they say there are 7 times 19, that's 133 elements. You take those 133 elements and express what the net excess of rightward elements there are. Okay, there could be a net excess of left elements. That would be negative numbers here. They z-score them across all the conditions, and you get this nice function. Well, it turns out that it's very easy to fit that function with a snapshot model. The idea being that at any one, the animal could look at any one of the 7 pulses, and if it so happens to this fit says that if 3 or more of the elements are moving, choose that direction. If you happen not to see 3 or more on that pulse, you would just take a guess. And that does a nice fit. Okay, the extrema model also fits it very nicely, and you can fit a diffusion model to it as well. And that would assume integration out to about 3 or 4 on average. So the point is that here's a here is a case from literature, and I don't really mean just to pick on this particular paper. You can't see what the paper is anyway, which is probably maybe a good thing. But one has to be careful about drawing conclusions about what a neural correlate of a decision is by simply asserting that that information is being integrated. So, for example, in the paper I'm thinking about right now, the assumption is that there's a simple step in the activity of the neuron that leads to the decision. Well, there ought to be a step if one is making a decision based on a discrete random snapshot of data. Okay, and I would say I'd say that basically I can't think of an experiment in the rat and mouse literature that looks at decision making that presumes counting over oftentimes as much as 2 seconds that isn't vulnerable to this critique. Okay, so we have to be careful. Okay, so how do you know your subject is integrating? Well, choice alone is not enough, and even with a psychophysical kernel, it's not enough. Okay, so you don't really know that an animal's integrating unless you test it. And the way you have to do that is you have to measure choice and reaction time. And ideally, you would do this by either by measuring a psychophysical kernel to rule out extrema and maybe to confirm or corroborate non-decision times, or I should say, relate reaction time experiments to which are sometimes called free response experiments, to experimental controlled durations. That is how accuracy improves as a function of duration. Okay, so that's the end of that section. And now I'm going to end ahead of time, it looks like, or maybe just on time, depending on how long I take now, is that I want to discuss something even more controversial and a little bit less insider, perhaps. And that is what knowledge is in the brain, what I'm going to describe as an intentional as opposed to a representational framework. I would guess that most of you assume that the job of the brain effectively is to represent information in such a way that the brain can then classify it. This is, I think, reasonable, although I really resonated with some of the things that Josh Tenenbaum said earlier today about thinking, or even the expressions of what we do with our physics engines were all sort of framed in the idea of sort of interrogating about a possible future state, or even, which I would say is like, what would I do with my hand? Let me get to that. Okay, so in these experiments, of course, the perception these perceptual decisions are being communicated through an action, and of course we've recorded, as I've been talking about, from neurons that associate information from mainly vision to information about what you're going to do, what the animal's going to do with its eyes. Now, of course, if we're recording from the parietal reach region, which is just across the sulcus from LIP, the area I've been focusing on in the first part of the talk, then you find that these neurons don't project to the frontal eye field. These neurons project to areas of the brain that control reaching. So if an animal were reaching to give his answer, you might expect these neurons to then evolve, and the representation of an evolving, or the representation of accumulating evidence bearing on the proposition, where will I reach? Okay, it's a proposition about left versus right, but really it's a proposition from the brain's perspective of what am I going to do about it, reach left or right. And lo and behold, that's the case. We found that. And it turns out actually the parietal reach region and LIP are active sometimes simultaneously. So there's actually a parallel organization of information flow in this intentional framework in the brain, at least when it comes to these very simple perceptual decisions. Now, to extend this idea a little further and get out of decision making per se, if we go to this area AIP, the anterior intraparietal area, it receives information from other parts of the visual cortex, not necessarily interested in motion per se, but it projects to structures in the prefrontal cortex that care about, that control the posture of the hand when we are going to grab something. Now, if you think about it, the idea here, and if those of you that know anything about neurology recognize that if you lose the parietal cortex, it's not that you lose vision or touch and ability to move, but you lose the ability to make sense out of vision and touch, and oftentimes the sense of our body is. And so the idea is that knowledge about space is not really the representation of where things fall on our eye or our visual cortex. Knowledge about space is about what we might do with that knowledge. We might reach here, look here, and so forth. How much time do you think I have?
3 minutes.
Well, I'm at 38 here, but all right, fine. I'll zip. Okay, so you've all learned about where and what pathways. Mel Gooddale replaced the where pathway with a how. I would claim the whole brain is a how pathway, okay? So this is just that ventral stream hows are more like affective hows. They're not questions about will I reach for this, will I look here, will I hold my hand a certain way. They're questions about will I eat this, will I explore this further, will I come back to this if I've explored down this one path, might I come back to this other path. There are questions about maybe will I mate with this, okay? So this is the point of information in the brain is not to have a label on it. It's to provide what I call a provisional affordance. Affordance being a Gibson term about what we might do with something, okay? And provisional meaning we don't have to do it. That's just the structure of knowledge in the brain is the organization in accordance with the kinds of things I might do, okay? And of course, that lives, it's structured as an interrogation, I want to emphasize, and that's a bit, I think, resonates with some of the language that Josh used when he talked about physics engines. And it has persistence. It has temporal thickness, okay? It doesn't keep up with a changing world or a changing body. It keeps us, it has a certain longevity, okay? And in the task, we've been talking about that longevity is on the order of hundreds of milliseconds to seconds, but you can imagine that there may be mechanisms that give us more longevity than that. So knowledge, I would argue, is organized as a provisional affordance in the brain, not a categorization on a representation, okay? And this is where the analogies and what we're going to learn, I'm going to be very provocative now, that from deep learning, machine learning, is basically going to be misguided if we care not about how to drive cars with these things, but how we drive cars with this thing, okay? And that matters to me as a neurologist, but it doesn't have to matter to you. Okay, so now, Jan. Okay, so now to end then, I'll just say that even knowledge in the abstract is a provisional affordance. The way to think about that is that when we make a decision about leftwardness and rightwardness, as a monkey has a very hard time doing, we find. But if you do, you can just imagine that the bigger brain just allowed a decision not about what to do next, but about what question to ask next. I'm structuring that language. It sounds like it's just information, but I'm structuring it saying in the brain, the strategizing or cascading out decisions is a way to think about an organization of a structure of behavior, I should say, that's organized as if we were planning the next act. It's just that we're just planning the next interrogation. And that's why I think that understanding this brain here, this little part of this brain even, of the monkey, has a relevance to the very complicated thought processes that our brains are capable of. And ultimately, I think what we need to do is to unravel this in a much more refined way in animals that may not be capable of these tasks, but have a biology that is more amenable to us. So I'll just throw these up here, and we can just leave it up here for discussion. Basically, persistent activity is a substrate for computation. Interpretation of neural activity relies on knowledge of the animal's strategy, and I was a little preachy about that, but we have to be careful about asserting integration and not testing it. And this, I just want to leave you with this, that persistent activity is propositional, intentional, and it's not really representational. Obviously, it involves representations of quantities and so forth, but it's not that we see things because they're represented. And we're bothered that we don't see things that are represented, like the gorilla walking on our visual cortex, so to speak, and invent strange concepts of attention in order to justify that commitment to representation. But rather, this propositional activity is organized as a provisional affordance, or knowledge is organized as a provisional affordance, I mean to say, and that this structure is what distinguishes biological intelligence from artificial intelligence. And with that, I'll thank you for your attention and point out that I got a tremendous amount of help with this talk from Gabe Stein and Ariel Zilberberg.
So we have time for a few questions.
You described, I think, the LIP firing of neurons as sort of like working memory, but that sounds very sensory, not very affordance-like. So could you describe the firing of LIP neurons more as a plan to move rather than the sort of the after image of where the field of the spot, where the spot was.
Okay, you wanted me to repeat the question. Is the LIP activity working memory, plan to move, but I wanted to go beyond that effectively. I wanted to say there was more than that. What the slide said is that it was discovered in the name of working memory and motor planning, okay? And I'm not saying it doesn't do that. I mean, that's how our screening tests effectively are working memory and planning eye movement. Those two things are confounded in that working memory task, right, of the eye movement. What I'm saying is that that persistent activity is a substrate for real computation. I didn't show you this, but if I showed you single trials from tasks where animals are reasoning from symbol, you'd see the response changing levels in single trials, not jumping to one number or another, but changing to intermediate levels of belief going along the way. So it's a substrate for computation. And it is, I think, the nature of the substrate. It is what is at the basis of the nature of what we call thinking.
So we'll take 2 more questions.
Right. Hi. Thank you very much for this beautiful talk. I was wondering about the intentional versus representational controversy that you want to raise. I was wondering whether you had multi-unit activity that you were recording from LIP, and whether this, so the data that you show us is mainly coming from single cell, right? Do you have multi-unit activity that would suggest that the activity in LIP is content specific with regard to the decision that is being accumulated?
So the question, I don't really know what this has to do with. You asked about multi-unit and single-unit activity. Most of our data is from single units, and then we would average, but we do occasionally get many units. And these days we use these probes that give us lots of units. So, but the context. I think you're asking a question about whether or not the neurons are context sensitive. I mean, we've trained these monkeys to see these random dots as bearing on the significance or the possibility that this thing or this thing will provide a reward. So in that context, we see these responses. I'm not really sure. I mean, if objects are present in the field and they flash on, I think that's an automatic invitation for the animal to interrogate them. I think that's what seeing is. It's the possibility of looking to something. Okay, so in that sense, that context, or maybe this is why you prefaced your question with intentional framework, is that in that context we can see where those interrogations kind of come from in the first place. But oftentimes they have to come from within because of a problem that we're trying to solve, and it's just not coming from a flash in the world.
Since you can always potentially act on some piece of information in the brain, how would you distinguish an affordance from information? How could you falsify the intentional stance?
I mean, the real predictions from the intentional stance, I think, come in the form of hysteresis in perception. In a way, I was too nervous to really pay as much attention as I wanted to that last talk. But there are many aspects of what we see or think we believe we see that I think, according to this intentional framework, post-diction would be a good example of it too. I've been trying to think of to falsify it. Let me think about that a little bit. I mean, to me, it's more of a philosophical position that allows us to interpret information in the brain in a sensible way. It's not. I'm not ready to engage in a philosophical discussion about it, but I'll think about that. It's a good challenge.
And we'll have to move on to the next session. Let's thank our speaker again.